I hate to be that guy, but accuracy is very important to me. Is the brain really meat? How many of you have ever had a juicy steak? Yeah? Recently? How many of you have eaten brains? Yeah? Do you remember what it was like? Did it seem like meat? It was strange. So I remember when I was a kid, I did taste brains. My mother cooked and served us brains. And I don't think it was much like meat. But I wasn't sure because it was a long time ago. So I consulted Google. I asked Google, right? And Google said that cooked brain has a soft, creamy, almost custardy texture with a waxy quality and a mild, fatty flavor that is often described as sweet or umami rich. After cooking, it can look like scrambled eggs or a rich, tender cloud, especially when fried. AI overviews can be a really good feature of Google, I discovered. So you don't have to take anyone's authority for it. Just try eating brain. It doesn't seem at all like meat. And indeed, it's not. So remember, our bodies, our brains, are mostly water. But if you take out the water, the dry weight of the brain is 60% lipid molecules. Your brain is mostly fat. Your muscles, on the other hand, are 80% dry weight is protein. Very different. Your brain is the fattiest organ in your body other than your fat, right? And so this is a fundamental difference in the structure of the brain. Why is the brain so fatty? That's what I want to start with today, the question. And to understand that, well, you have to know a little bit about what the brain is. And the journalists tell you your brain has 100 billion neurons, your brain has thousands of synapses per neuron, but I don't like those factoids as much as this one. So consider that the branches of neurons, they're kind of like the wires of the brain, right? You've seen pictures of neurons before. They don't look like ordinary cells. They have these branches. And if you took all those branches and you laid them out end to end, how long would that reach? You have any idea? So the estimates are millions of miles, right? Round trips to the moon and back. Your brain is almost completely wires. It's these entangled branches of neurons, all stuffed inside your skull. And the wires, well, they're not really wires. They're wires in quotes. They have, our electrical wires have copper. The wires of your brain have salt water as the conductor. But they do share something in common with the electrical wires, which is they have to have insulation. You need insulation to keep all those electrical signals separate. And that insulation is the fat, right? That's why your brain is so fatty. What's that? F-A-T. Fathead, it's an insult, but it's the reality, right? And so this is, some people might say, oh, I'm telling you something really important about the brain. They might say, well, Professor, we already knew that. We already know about neurons. We know they have these branches, and that was discovered in the 19th century. So what are you telling us that's really new? So I'll make a comparison, right? So everyone knows about the double helix, right? Watson and Crick, 1953. They figured out that DNA has this helical structure, and the information of heredity is encoded as letters in this, a sequence of letters in this double helix. But that tells us part of what the structure of DNA is like. To really know the structure of DNA, we got to know those letters. And that was accomplished 50 years later by the Human Genome Project, right? 3 billion letters of the DNA sequence. And 50 years, it's kind of long, but it's also kind of short. Watson and Crick were both alive to see the fruition, right? To see the actual sequence of DNA revealed. But the brain, a much harder nut to crack. It's true that in the 19th century, we already knew that the brain is wires. We have this complex network of many neurons inside the brain. But knowing that there's this network and knowing that there's wires is not the same as mapping out this network, right? Mapping out this network means tracing every inch of those wires. And that's hard. And we still haven't done it for a human brain. We wanted to do it for a human brain, but it was too big. And we wanted to do it for a dog brain, and we wanted to do it for a frog brain, and we wanted to do it for a fish brain, but a fly brain, still too big. Fruit fly brain. Yeah, well, that's maybe the right size. So I'm going to show you what is a fruit fly brain really like. I mean, you may not even have thought about it, but flies do have brains. And this is a kind of a joke. This is my collaborator, Mala Murthy. She's the director of the Princeton Neuroscience Institute, and we led a consortium that mapped out the fly brain, and this is our joke about how it started. So I'm going to show you what a fly brain looks like. Imagine we have a fly brain. If we zoom in, at really higher and higher magnification, you can kind of see now the structure of this fly brain. These are the optic lobes on either side. They're like Mickey Mouse ears, and they're right behind the eyes of the insect, right? And so we zoom in a little further. And now we're really zoomed in. We've zoomed in by 10 to 100,000 times. These are images from an electron microscope, the world's sharpest microscope. And it's a 3D image because I can go from slice to slice like this, and we can see slice by slice what a fly brain looks like. And this right here, I found one. This is a synapse. So you might have heard about what a synapse is, right? A synapse is a junction between two neurons. It's where two neurons communicate with each other. They send signals to each other. And on the sending side of the synapse, there are these round things. They're called vesicles. And they contain molecules called neurotransmitters. You might have heard about acetylcholine, glutamate, dopamine, serotonin. All those things are contained inside vesicles at synapses. And so when the sending neuron wants to send a signal, it spits the contents of those vesicles outside, and they cross over the gap to some other neuron, and that neuron can sense the neurotransmitter molecules. That's how the signaling is happening. And you may have heard about a synapse in abstraction, but this is what they actually look like if you could use a really, really powerful microscope. And so I tell my undergraduates that you might be watching a Puccini opera or watching your TikTok feed, but either way, your neurons are just spitting on each other. Or if I'm feeling more lyrical, I will say, they're spreading their perfume to each other. right? So that's what a synapse looks like. But I also told you about the wires. Where are the wires? What you're seeing right here is the outline of a wire. Imagine I took some spaghetti and I chopped it up. I took spaghetti and I made it hard and I chopped it into a salami kind of. Every one of those strands of spaghetti would have a cross section, and that's one of these cross sections right here. So to trace the paths of all those wires, we have to go slice by slice and follow one of those contours. It's like a 3D, enormous three-dimensional coloring book. Color, but stay inside the boundaries of a neuron. And if a single person were to do this, we estimate it would take about 100,000 years to color in every single wire. But luckily, we have AI. Artificial intelligence can look at those images and do the coloring for us. So I'll show you. Here it is all colored in. Slice after slice after slice. And if we zoom back out again, if I click on any one of these neurons, what we see is the actual neuron colored in 3 dimensions. No matter where I click, we get another neuron. All right, so you've probably seen pictures of neurons in isolation, but it's completely chock full of neurons. So we just zoom back out again, and we see again. There it is, all colored in. So if we could trace the path of every wire, find all the locations where they talk to each other, we have a complete map of the connections in a fly brain, and that's called a connectome. And a connectome doesn't exist for any large brain. It's only been complete for small brains like this. And this connectome actually was released only last year. So you're one of the first people on Earth to actually see this kind of mapping done for the first time. Okay, so we did this. And one kind of analogy you might think about is that a connectome is like the map you see in the back pages of airline magazines, right? You have cities and flights between cities, but just imagine for a fly brain, it's 100,000 cities, it's tens of millions of flights, so it would be a really thick airline magazine. And we got a lot of media attention, right? Old media, influencers, shout out on Saturday Night Live. I finally did something to impress my colleagues in computer science. But you could ask, so what? You have this crazy map of a fly brain. What do you do with it? Well, one very basic thing that scientists do is just make catalogs. It sounds kind of trivial, but if you were a 19th century naturalist, you might make catalogs of the different kinds of bugs in some tropical forest, right? And so we can do the same thing with neurons. A layperson says neuron, but to a professional neuroscientist, there's many different types of neuron. And I'm going to show you those types of neuron here. These are the 230 types of neuron in the fly visual system. Over half of the fly's brain is neurons dedicated to vision. They're very visual animals like we are. And those, let's say there's 40,000 visual neurons on one side, they can be classified into one of 230 different types. So it's kind of like botany, right? We see all these wondrous forms. All right, so what? You cataloged all these neurons. What do you do then? So I'm going to quote from a son of Boston, Louis Sullivan, historic famous architect. And he wrote that it is the pervading law of all things organic and inorganic, of all things physical and metaphysical, of all things human and all things superhuman, of all true manifestations of the head, of the heart, of the soul, that form ever follows function. This is the law. So he was preaching about this idea that form and function are related. And so one might ask that question. It's often the case in biology that form and function are related. If you see a heart, it's not hard to figure out that a heart might be a pump, right? Form and function are related. But is it true for neurons? And so I'm going to show you some examples. I'm going to show you examples starting out with the visual system of the fly. And I want to remind you that a fly has a compound eye. It's not an eye like ours, but insects have these eyes that are composed of lots of little eyes. In this case, the fruit fly has 800 little facets of a compound eye. And this cartoon is funny, the last thing that a fly ever sees. It's funny because it's not true. So it's not that those little eyes see a whole image. Every little eye is seeing 1 pixel in the entire image. So if there's 800 little eyes, there's 800 pixels in the image sensed by the fly eye and sent to its brain. But this is kind of a fun idea too. All right, so T4A cells. This is one of the types of neuron that was in that video. And you could see the fly brain right here, and this is the right optic lobe. So here we are. I'm going to zoom in. It's beautiful. You can see these cells. So I'm not showing you all 40,000 neurons. I'm just showing you the 800 neurons in this fly visual system that are T4A cells. And there's one of them per pixel. So there's not just the eye that has the structure. Inside the visual system, there are some neurons that are just one for one with the pixels of the fly eye. And so there's 800 neurons here. But and this is all false color, right? The real fly brain doesn't have these colors. They're just colored in by computer graphics. But the forms are real. They came from real data. You can't see much. So what I'm going to do is I'm going to hide all but a few of those neurons. So they all have a very similar appearance, right? And then I'm going to ask you to look at something, which is that, look at these neurons. Well, I'm going to actually give you an analogy here, right? Most trees are symmetric. They have a crown that extends in both directions. But there are trees that are very asymmetric for whatever reason. And if you look at these neurons, they are all going this way, right? All of them have their branches sweeping off to one side. So they're like this. It's a great question. So is there nobody knows what causes them to grow this way. But it's not as simple as that because there's T4A, T4B, T4C, and T4D. They look very similar. A does this, B does this, C does this, D does this. So there must be 4 different winds for those 4 different neurons. Nobody knows exactly why. Now, why do these neurons do this? What does this mean? Well, what I can tell you, when people, neuroscientists, researchers, and I want to make clear that this is not my work. I'm speaking for scientists in general, right? I'm talking about the work of many people. So neuroscientists can observe the activity of neurons in a living fly brain, right? I mapped I worked on, my consortium, we all worked on mapping a dead fly brain. They can actually look inside the brain of a fly and see the neurons being active. And what activates these neurons is moving stimuli. You know, a fly has to see motion, right? If you're going to it with a fly swatter, it has to see motion. And each one of these neuron types is activated by motion in one of 4 directions. And in fact, this T4A neuron is activated by motion in that direction. It went that way. It's kind of crazy, right? The form somehow seems related to the function. And in my fanciful way, I think these neurons have been buried inside that fly brain for 200 million years. They've been trying to tell us something about what they do. And you're among the first people who've ever seen their pantomime. All right, and I'll give you another example. And this is not from the visual system of the fly. So you can see the optic lobes are here. This is called the central brain. These are called the EPG cells. And let's zoom in. There's 50 of them. Now, this is really crazy. There is a donut at the center of the fly brain. Right? I mean, could you make that? Who could have made? Nobody could even imagined that there is a bunch of neurons that form a donut. Yeah, I'm just showing you 50 neurons out of 100,000, right? They're actually making up a donut. And what do the individual neurons look like? They are making up wedges of that donut. What could that possibly be doing? Well, again, researchers have observed the activity of neurons in living flies, of these neurons. And it turns out, well, make an analogy. You've used Google Maps, and it tells you where you are, right? But sometimes it also has an arrow telling you which direction you're pointing. These are the neurons that compute the direction in which the fly is heading. And it makes sense, right? Because direction is 360 degrees. So yeah, let's make some neurons in the shape of a donut, and they'll encode an angle. And that's real, right? That's reality. I don't think I could have made that up, right? So this is really an amazing, again, an example of Louis Sullivan's idea of form and function. Direction in which the fly is heading. Many animals have to navigate. They got to know the direction. That is encoded in a donut-shaped structure in the fly brain. Okay. Now, I've told you about, why is that? I don't have time to explain why that's the case. But the reason is that a neuron's form actually constrains its connections, right? Where a neuron stretches its branches to determines which other neurons it can connect to. If I don't reach out and touch someone over there, there's no way for me to connect, right? So form constrains connections, and it's become a familiar idea in artificial intelligence because AI these days is based on artificial neural nets, that it's the connections of a network which determine its ability to function, to do computation, to do all the operations of intelligence. Connectionism. All right, so I don't have time to explain that, but I've given you a flavor of the idea of form and function. Now you could ask, can a fly think? Right? So for the visual system, it's all about encoding the properties of the visual stimulus. Is it moving or not? But the researchers who discovered this donut, they argued it's kind of like thinking, because if you turn the lights out so the fly can't actually see anything, that donut still maintains activity that encodes a direction. And if you rotate the fly imagine that you're like this, your eyes are closed, and you turn this way, you have an idea of which direction you're heading. The activity pattern rotates around the donut while the lights are off. So this means that that activity pattern is not encoding where, which direction the fly is going. It's about which direction the fly thinks it's pointing. So it has something to do with thought, right? Thoughts are mental activities that are not directly related to what you're sensing right now, like my students during my lectures. It's not directly related to their input that I'm giving them, right? They're thinking about something else, right? That's thinking. All right. Well, of course, you have to ask, does a fly have a soul? Well, I'm going to invoke an authority figure here, René Descartes. René Descartes grappled with a question. He believed that there's a soul. He's a so-called substance dualist, right? He believed there's a soul and there's a body. But while you're alive, the soul has to be tied to the body. Well, obviously, it must be in the brain, right? Soul must be tied to the body and the brain. But where in the brain? He thought deeply about this. Where in the brain is it that the soul resides? And so I'm going to give you a quote. He found this little red structure in the brain called the pineal gland. And he said, this is the seat of the soul. And his reasoning actually was pretty interesting. Because you see only one thing. You have two eyes, but you see only one thing. You hear one voice, even though you have two ears, right? You never have more than one thought at a time. So it can't be, the soul cannot be in any structure that is on both sides of the brain. It has to be on a structure that there's only one of them. And that's the pineal gland, according to him. And so I don't know if a fly has a soul, but if it does have a soul, it's got to be in that donut. Right? It can't be in the optic lobes. There's two of them. Kind of makes sense, right? I mean, when you're navigating, you don't think you're going in two directions at the same time. You have to go in one direction. So, having brought up the idea of the soul, I guess I thought about Stephen Hawking. I mean, everybody knows Stephen Hawking, one of the most famous 20th century scientists, and one of his most memorable quotes.
And I quote, "If we find the answer to that, it will be the ultimate triumph of human reason, for then we would know the mind of God."
Wow. Stephen Hawking, he said that the goal of physics was to know the mind of God. You know, I was trained as a theoretical physicist. That's my PhD. Over the years, I've become more humble. I just want to know the mind of a fly. I'd be satisfied with that. Of course, those of you who are religious might say, well, having seen the complexity of the fly brain, which you never imagined before probably, you might say, still, I'm studying the hand of God. So just for fun, I want to show you one last video. This is a science influencer.
It's taken over a decade, but we finally have it. A completely mapped fruit fly brain. This is significant because it's over 140,000 neurons all connected together, the equivalent of over 500 feet of wiring. This is the first time a full map of a brain and its connections has been done for a complex animal brain. Researchers around the world are going to use this forever and for other animal models. But what really blows my mind is that they were also able to put this brain into a program. They were able to take all the connections and make it like a computer program. And then when they gave commands to that brain, like there's food there, the simulated brain said, well then move your mouth closer to it. This means animal brains can be replicated in silico. It also means that probably yours can, which also means that probably free will doesn't exist, and there's nothing apart from you from your meat, and that the afterlife.
I showed this video to my family and I saw the generation gap. My wife said, gross, who is this guy? And then my daughter said, wow, he's really cool. He was talking about one of the serious scientific papers in the special issue of Nature in which the fly connectome was published. And this paper took the connectome, this map of connections in the neural network inside the fly brain, and attempted to build a computer simulation of that fly brain and give it a virtual stimulus and see a virtual response. And so you may have seen science fiction movies about the idea of mind uploading, the idea that the information in your brain could somehow be transferred to a computer and a computer simulation of you could be built. So mind uploading is, of course, science fiction, but mind uploading for a fly is now mainstream science. It's serious science. It's non-trivial because the connectome is an incredible stepping stone towards realistic brain simulations, but it doesn't give you all the information you need to make a simulation. So I'll leave it at that. People are working on this problem of brain simulation, realistic brain simulation. Sometimes we use the term brain emulation. And this, of course, raises a lot of deep questions. The idea of building a realistic simulation of a brain is really thought-provoking. And I'd like to joke that one fly died for that fly connectome, but this is the first fly that will live forever in simulation. All right, so I would like to just wrap up then here, and I want to thank the FlyWire Consortium and the incredible fruit fly neurobiologists whose work I've been talking about this whole time. I think a lot of exciting things have happened. I'd like to thank Amy here, Executive Director of iWire, and she's been helping out with creating the great visual assets, and also Tyler Sloan, a visual artist. Some of his work I showed here, and he'll be, some of his work, his science visualizations are being shown on these monitors here. And I'd love to thank all of you for coming and enjoying the breaking news in science. Thanks a lot.
Well, thank you so much, Sebastian. And if you want to ask him a question, please go to slido.com, type your question in with hashtag FlyWire. So what I learned from your talk is that the soul might be a donut in our heads, which makes sense here in Boston, right?
But is it a Dunkin' Donut or a Krispy Kreme is the question.
It might be a donut hole, who knows? So let me get back to this idea that you ended off with. I mean, you started with this really fascinating video of, I think, like these two inorganic beings talking about they're made of meat, they're on Earth. And you might think of like a computer-generated like intelligence. And it's so fascinating how your talk circled back at the very end toward the possibility of, at least for the fly, uploading the fly brain, the connectome, virtually. So my question for you, Sebastian, is, my gosh, what have you done? Where are we going with this? Because, right, it would seem possible that if we ever get to the point where of a human brain connectome, one would want to do something like that in silico reproduction. Yes.
Well, we're not there yet, but of course, one of the reasons I was reminded of that old story is all of the current discourse and chatter about artificial intelligence right now. Sometimes when you hear the tech people talk, it's like, oh, they talk as if humans are obsolete and electronic brains are superior and so on and so forth. And so that's the kind of joke of that short story, right? That the aliens can't imagine an intelligence that's based on biological matter, right? They can only think of electronic intelligence. And so it's very much the zeitgeist. Now, but you're right, the question of simulating a brain, I think, is kind of dual to the idea of creating an artificial brain. Artificial intelligence and studying brains and maybe reconstituting them in silicon, they're related to each other, these ideas.
Well, we have already plenty of questions that have come in. So let me start with this one. Is there a way to describe the complexity of the brain mathematically? Can you demonstrate how this complexity changes with age or repeated toxic exposure? There's a lot you could do here.
Well, that's one of the basic ideas about the brain is that as we develop, as we age, or as we learn something new, some change is happening. And it's an old idea that it's a change in the pattern of connectivity in your brain, the connectome. And one of the blockers in making that into real science, as opposed to theories and indirect kind of speculations, is that we haven't been able to observe the connectome. We haven't been able to observe the thing that is hypothesized to change. And so that's, in fact, why I and other people in what was formerly kind of a cult, a cult within science, we believed that the connectome was key to making real scientific headway on these kinds of questions. Now, whether it could be done mathematically, well, we have no choice, right? When we have complicated networks and huge amounts of data, then a mathematical and computational approach is necessary to make sense of it. I showed you pictures, and there's some things which I can explain through pictures, but some things which I can't.
This is a really good question. How are you able to tell the function of a neuron if the fly brain is dead? Is it the location that's the main indicator?
Well, I didn't, I tried to argue that the function of the neuron is hinted at by its form, right? So the neurons that detect motion in a living fly, in a dead fly, I can see that they're all pointing in one direction. And in fact, the direction in which of the moving stimulus that they like to respond to, right? Or the donut neurons, right? So somehow the form appears to be related to their function as measured in a living fly. But ultimately, it's not just the shapes, it's the connections. And so one of the goals of brain simulation is to build models of neural nets with these connectivity patterns that these neurons actually make up in real brains, and try to replicate the function in a simulation. This is more like looking at the patterns, right? Looking at a heart, seeing that it has chambers and it has all these valves and so on, and inferring that it has a pumping kind of function.
This is so I'm getting to questions now where people want to then kind of discuss the connection, no pun intended, the connection between this work with the fly brain and maybe what we might learn about the human brain. So here's one. Does the human brain have the same kind of neurons? Are they the same? Are they different? What can you say about similarities and differences?
Well, the most obvious difference is that human neurons are huge. They're enormous, right? So a fly neuron has to fit inside a fly head. And we're used to thinking of neurons as cells and therefore microscopic. But the crazy thing is that they are kind of microscopic in some way. There's a nucleus, a round part that is small. But the branches of a neuron they often stretch from one side of the head to the other, right? They go from the brain down to the spinal cord. So a neuron is not really a microscopic object. It's a macroscopic object. It's huge. It's enormous. And so that's a very big difference between a fly brain and a human brain.
Here's a little plug for the museum. As you were walking in, you've passed by this big blue neuron that's projected up on the rear lobby screen. That was from the Jeff Lichtman lab at Harvard University. That's a real human neuron taken from an epilepsy patient during surgery. So that gives you an idea of just how complicated that human neuron looks like.
But at the same time, it's surprising, right? So our evolutionarily speaking, our ancestors, it's hundreds of millions of years ago that we diverged. We're very distant relatives of flies, right? But acetylcholine, dopamine, those things are actually found in fly brains too. So there is a kind of unity of all life, right? At the molecular level, there's an amazing unity of life. The way that synapses are not exactly the same, but the way that a synapse works, there's a lot of commonality between a fly synapse and a human synapse.
Another question. Excellent work. How do you see this being translated to understanding the human connectome? Ultimately, the holy grail of how our brains work.
Well, the first thing I would say is that neuroscience is at a very primitive stage. So if we can understand how even one brain works, I know everybody wants to understand the human brain, but if we can even understand how one brain works, that tells us something about how all brains work. Because when your ignorance is enormous, just a little bit of knowledge often tells you something about all things. It's only when you know a lot that what you learn from a particular instance only tells you about that instance, right? So that's the first thing I would say. But in terms of, it could be that we don't know how the future will unfold. It could be that by the time we understand how human brains work, many of the basic principles will have been worked out in small brains. What's the difference between a human brain and a fly brain? There's some maybe fundamental differences, but there's also there's scale. There's a lot of neurons in a human brain, right? And in AI, we know the idea that when you scale up the artificial neural network in AI, completely new capabilities emerge. So part of it may be the same principles, but a different scale. Nobody knows yet.
Tell us a bit more about the FlyWire project. So there was a very strong citizen science component to that. Was that essential for the work? And please explain what that connection was with citizen science.
Right. So our earlier project, which Amy is the executive director, it was called iWire. It launched in 2012. And so what that did was enlist humans to help color in the neurons. I showed about coloring in the neurons, right? In the meantime, AI has gotten much better at coloring neurons. So what the humans are, of course, still fixing the errors of the AI, but there's a lot less fixing required. So the biggest contributions actually of the citizen scientists in FlyWire were not the coloring of neurons, although they did a lot of that. The most remarkable contributions were in classifying, cataloging the neurons. It's kind of like bird watching, right? Amateur scientists can help catalog different animal species. And so in the same way, our citizen scientists did a tremendous amount of work in cataloging the neurons that I showed before.
So we have time for just a couple more questions. You had initially mapped out the brain of a male fly. Was it male?
Female fly.
Female fly. And another group did the male fly.
Recently, in fact, just a week or two ago, the connectome of the male fly was released. That's right.
Were there differences in the two connectomes?
Ah, yes, male and female. That's what everybody wants to know, right? Yes, so there are types of neuron that are only found in the male fly brain, but not in the female fly brain, and vice versa. So there's sex-specific neurons. And this is something that can be conclusively demonstrated in small brains right now, like flies. Now, whether that's true in the human brain, nobody knows.
There's so many excellent questions. I hope you'll get a chance to chat with Sebastian during the reception, so I'll have to just end with one last question. What project are you working on next? What are people trying to do in terms of other species for mapping out the connectome?
Well, it's a menagerie, right? There's different insects, there's fish, there's mice. People are doing, what we developed were foundational technologies. In some sense, I don't have to make any discoveries, right? I'm an enabler. So I work on making it possible for other people to discover things using connectomes. So people are happily applying this technology to all kinds of different brains. But personally, I'm involved in, I want to make all this faster and cheaper so that everybody can use it, all the researchers can use it, and have the maximum impact possible. And I'm personally also very interested in simulating brains. So I started out as a person who built brain models. And now that we have these wiring diagrams, these connectomes, I'm returning to the question of, can we simulate a brain in a realistic way?
I told Sebastian on a call leading up to tonight's event that after seeing his data and his videos of the fly brain, I feel kind of bad now when I try to swat a fly. So I hope that you've enjoyed the
There's
presentation.
plenty more where that one came from. So,
But they're so intricate. I feel a little bit bad. So thank you so much for joining us and for giving us your Albright lecture. It was really fascinating. Thank you so much for your time.
thanks a lot, Insoo.